Last but not least, we have our steroid receptors. With our steroid receptors, these are things that are going to bind our steroid hormones and hormone-like compounds. We have two different pathways that will be involved with this. We have the NIS pathway and the MIS pathway. The NIS pathway is a nuclear-associated or nuclear-initiated steroid signaling, which means it's going to affect the nucleus, and when it affects the nucleus, that means gene transcription, which is really slow. So when we're looking at changing signaling pathways or things like that, the NIS is not involved with that at all. With our steroid receptors in the NIS pathway, which are involved with regulatory genes, we have our ligand binding domain, which is our C-terminal. We have our DNA binding domain. And then we have our gene regulatory, which is the N-terminal, which is going to tell it to go ahead and turn on. We have type 1 receptors and type 2 receptors. Our type 1 receptors are going to bind our steroid hormones, and they are made from cholesterol. Cholesterol has a steroid nucleus. It's a 4-ring nucleus, and then cholesterol is going to be converted into the common precursor, which is progesterone, and then from there we can bud off and make different steroid hormones depending on which cell that it's in. We have to make progesterone first. Your type 1 receptors are located in the nucleus, and they're not found in the nuclear membrane or anything like that. They're within the nucleoplasm, and it's going to bind estrogen and progesterone. In particular, if type 1 receptors are located in the cytoplasm, not within the nucleus, it will bind androgens, so like your testosterone, and our glucocorticoids. That would include your cortisol and your aldosterone, which aldosterone is important for blood pressure regulation. So when we are in the NIS pathway, nuclear-initiated steroid signaling, again we're looking at transcription, your type 1 receptors in particular are going to bind to a specific area on the gene, which is referred to as the hormone response element. So it'll bind to this HRE, this area on a gene that's going to go ahead and tell us to translocate. So it tells us to go ahead and stimulate transcription. Turn that gene on, make more of it. Our type 2 receptors are located in the nucleus as well, but they will bind different things. That'll bind vitamin A, that'll bind vitamin D, and our thyroid hormones. Now with our type 1, there's no repressor protein bound onto it. Once our steroid hormones bind onto our type 1 receptors, it's going to recruit a co-regulatory protein, whereas our type 2 receptors have a co-repressor protein that's bound onto it, and then once our vitamin A or vitamin D or thyroid hormones bind onto that type 2 repressor or type 2 receptor, the co-repressor gets released, and then we will bind a co-activator. So we have a co-regulator with type 1, we have a co-repressor that's replaced by co-activator with type 2. The other pathway with our steroid receptors is membrane initiated. Steroid signaling, which is going to be located within the membrane, so plasma membrane, and in particular it's going to be located in that little cave, that calvuli, that cholesterol-rich region. And with our MIS pathway, it can bind to our steroid hormones, can act as ligands. Thyroid hormones and vitamin D can act as ligands as well. Vitamin A cannot. And the MIS pathway will not stimulate transcription primarily, but it's going to activate a whole bunch of those other pathways we talked about with G protein signaling, as well as catalytic signaling as well. It's going to activate signaling through activation of SRC or G proteins. It can stimulate phosphorylation pathways, so a lot of the protein kinase C with our phospholipase C pathway, protein kinase A. So with phosphorylation, but MIS can also interact with NIS, so it can also change those co-regulatory, co-activator proteins. So there's some crosstalk. So NIS is always going to, your NIS pathway, nuclear initiated, will always affect gene transcription. MIS pathway may affect gene transcription, but it's much more likely to activate some of those rapid pathways. So this is your fast steroid signaling, and your NIS is your slow steroid signaling.